surprise and expectation. And this goes back to the 19th example. There are people who study it and study the role of cerebellum in it. But if you go back to they simultaneously published in 1950 two papers. One of them called what they were studying coronary discharge. The other one called it copy. But it's sort of this is a very important idea. It's, to me, it's the most important thing about neuroscience I did not know, that I knew nothing about, and very few people talk about it. But it's one of the most important things that make it possible for us to engage in behavior, as it turns out. And it's been studied now mechanistically in a variety of different systems. It may be most familiar to you from stochastic systems. So, as you know, when your eyes are dark, this right now, your eyes are moving continually, and yet the world is not jerking around in front of your face. If it were, you would be continually nauseous. So you have a system that is basically keeping track of where your eyes are going, and then updating your visual field so that it stabilizes the image. That's what it means to stabilize the image. It's not really stabilizing an image anyway. It's keeping track of where your eyes are going and compensating for that movement to give you the sense that your eyes are not moving. And that can only be done through corollary discharge mechanism. So, if we just think about the two kinds of way, you have movement that others produce. Me shaking my own hand and you shaking my hand for me. Then we have movements that have corollary discharge associated with them. Now, that information we're getting from our limbs for moving them. And self and other. It's a distinction about what makes you and what makes everybody else. Now, this box can never be filled unless the SPA existed. Okay? Obviously. This box has never been discussed or filled. I will do my best to fill it, because what do we do with scientists except fill boxes? So, let me just get to the motor system produces motor output. Motor output, you have movement command of motor neurons that can produce a movement. At the same time, the motor system is sending out a signal down there. It's also sending out what we can call a motor copy, a copy of this command to another part of the nervous system, inside the nervous system. The afferents from that movement will then be compared with the corollary discharge to give a sense of whether or not you have an unexpected effect. So if I'm moving my hands like this, if I accidentally touch something, I will detect a difference between what I intended to do, or what I expected to do, and what actually happened. That gives you the ability to detect salient objects in the external world. So when you are an unexpected event, that's important for you. That is a signal that there's something out there in the world worth paying attention to. And so that different signal is then what is conveyed to other parts of the brain for this process. So using this model as our basic idea, we then I'll just cut to the big experiment we did to answer the question of can we fool a rat by moving its time limit away, where in one case it expected the outcome and in one case it did not. Okay? So the difference between by reporting brain activity at the same time, can we mimic what we see in the difference between a twitch and a wake? Okay? So how do we do this? Here's a rat, there's a brain, there's a spinal cord, there's a hindlimb. We produce what we call generalized arousal, which we interpreted, which expected would involve a corollary discharge signal. We're going to stimulate the animal's snout with a cold stimulus. It's very arousing to a rat. They have lots of thermoreceptors on their face. And other ones as well. If this were like a waking movement, any waking movement, we expect the local field potential of the brain and the motor cortex would not register in a vacuum. And that's exactly what we saw. So, here's the killer experiment. We made the spinal reflex, the local spinal reflex, which we presumed or we hypothesized would not involve a corollary discharge. It would be like an unexpected event. So what we did was we flipped the tail, and that produced a local reflex that would then move. And now what we saw, what we predicted was we would see an enormous effect. And that is exactly what we saw. So by manipulating the expectations the animal had about the movement that would occur, we were able to mimic what we saw in the natural circumstances of an animal being awake versus an animal. And this video shows how this plays out. You flick the tail, you elicit a response, a reflexive response of the limb, and you see really nice, robust activity in motor cortex. But when we stimulate the snout, we produce generalized arousal, the limb goes crazy, but we don't get any activity. So, to go back to Latin square, occupied in this box, twitch or react. It is a form of self-generated. It's a way of surprising yourself in a way that you think makes twitches a distinct class of movement that's being produced and has effects that are very distinct from what we do now. So the basic model is to go back to wake movements and how they have movement command and corollary discharge. In our case, we're lacking the corollary discharge because the system is acting as if there is no corollary discharge. We don't know exactly if that's the case, but it's acting as if there's no corollary discharge. And then the signal gets conveyed with high fidelity to other parts because it's not getting canceled out by corollary discharge. And then this raises an important functional question. Well, why suspend corollary discharge during twitching? And this is where we had a aha moment. Because all along we should have predicted it. If we were arguing that twitches are important for the self-organization of the nervous system, that's providing you with critical information about the structure of your limbs, the biomechanics of your limbs, how many joints you have, how many muscles, which muscles are connected to which, amplitude of the tendons, etc., etc. The last thing you would want to do in such a situation is to cancel out the information you're getting from the periphery. So if twitches were in fact important for self-organizing the sensory motor system, you don't want to have corollary discharge because you want to get that really clear signal about how your limbs are constructed. So that if you're Johnny Ack or you're Faith the dog or Duncan on the beach and you're developing without forelimbs, you're developing without hind limbs, you can actually figure out that you don't have forelimbs and hind limbs, learn about your body, and behave in an individual fashion. Which also leads us to the next question of where might this comparative be located, because no one yet has ever recorded a corollary discharge signal in an animal. So we haven't yet to do that. It's been done in chickens, it's been done in fish, magic fish. It's never been done in mammals, and it's kind of important to figure out where these signals are being produced. And one place that we thought could be very important would be really far back in the medulla, because we ruled out the cortex as being involved in this at all. And the external cuviate is a four-limb proprioceptive relaying or processing system that's one of the first brain areas. It's part of the dorsal column nuclei. It's called the medulla. So again, this is the same thing over again. And this is a caudal medullary recording of an unanesthetized fetal rat. So it's not the easiest thing to do, but what you can see is that the animal is asleep. Here in EMGs, the animal wakes up, and again we see the same pattern. It shuts off, but the animal starts twitching again, and we see this activity. And if you look at the period histogram for this, 0 is the place where the twitch occurs. You can see there's a very short latency response for this, showing that this is a response to the twitch itself. But we don't have a definitive answer yet, but we're getting close to determining what that one shows is that we're getting close to being able to assess the source of the corollary discharge and where the comparator is located. Search for the corollary discharge itself is getting us even farther into the periphery, looking at spindle, muscle spindle. That's for a whole other talk. I can't go into any detail about that now. But all of it led us to then look at really what's going on. Mystery, and the red nucleus, it turns out, is a very important area for looking at this. It's recorded now. We've completed the study of recording from this important motor area in the brain stem. And first, again, the sensory motor problem comes in because think about the red nucleus as a motor area, but it's also receiving sensory input. You can show that by stimulating the forelimb and reporting from an area of the red nucleus that responds to the forelimb, we see really robust activity following it. So the red nucleus is not just the motor nucleus. And you can see this nice following response after you stimulate the forelimb. So here's a film showing two different types of red nucleus activity. In this case, the unit of activity occurs in the red nucleus, and you'll see that the twitching follows, the unit activity precedes the activity that you see in the forelimb. Right? And so you can see that over here as this preceding response activity before the limb moves. Okay? That's what you expect in motor twitching. But here is a sign of what happens during twitching. Now you get a twitch, and then you have activity in the red nucleus that follows the twitch. You'll see the twitch right here, and you'll see the activity there. And here's the average activity showing that this red nucleus is actually following the twitch. So that is really interesting because we know about how information goes back to the red nucleus. It can go either through the cerebellum or directly back here. But no matter what's happening, what this means is that the red nucleus is a site of sensory integration. It's not just producing movement. Of course, it's not just responding to movement. It's actually integrating. We think it's integrating sensory and motor information. And we think that's important for the topographic organization of this nucleus, which we also need to exist. Let me just say a little bit about the cerebellums because it's so important to what we've been doing lately. Cerebellum is a great structure to study because it shows a lot of postnatal development. 13 days. The cellular structure of the cerebellum, and also in the use of Purkinje cells, shows just how rapidly they develop. We don't learn about the emerging circuitry of the structure and how exquisitely organized it is. So we're using this understanding of the circuitry to explore how twitching changes and how the effect of twitches on the cerebellum change as the circuit is changing. So we can actually relate those two things together. And again, as we've often seen, we see lots of activity in the cerebellum when the animals sleep, and then they wake up. It's almost becoming, it's like a broken record, but again, we see lots and lots of Purkinje cells that show nice reaffirmation by twitches. Why would you care? Well, you may not be aware of this, but early cerebellar injury is one of the best predictors of the onset of autism. So the first predictor of autism onset, the highest risk factor is associated with having an identical twin with autism. The second highest factor is cerebellar injury at birth. Okay. So now we're starting to get to a place where we can start to think about how early behavior, especially behaviors that we're studying during sleep, how this behavior can set in motion cascades of developing neural circuitry that make possible normal development, and when they are disrupted, can lead to developmental disorders. And it's really the past year, what they said was that normal processing of any type that affected necessary integration could be like cognitive development. So imagine that the cerebellum, which is intimately connected with the thalamus and association cortex, every area of the cerebellum has a topographic link with every area of the cerebral cortex. But imagine that, and the cerebellum really develops earlier than the cerebral cortex. Now you have some kind of a focal injury to the cerebellum. You could have a genetic mutation. You could have a stress issue. You could have environmental insult. Any of those things that one of the problem with the cerebellum could have organized relationships with other parts of the brain. And these sorts of things are thought to be involved in sorts of problems that you see in autism. It also can play a role in other disorders like schizophrenia and things like that. So I think this is just the beginning of understanding how early developmental events can have these sorts of effects. Well, our hypothesis is that sleep disruption or restriction early in life disrupts the development of the system with tragic consequences. And I don't need to go much further than just thinking about what happens in certain highly deprived circumstances. Such as what happened in Romania under the Ceausescu regime, where these human infants are being raised in these incredibly depleted conditions that you know all too well about. And so under these conditions, I asked Chuck Nelson, who did involved in these studies, what do you know about sleep under these conditions? And they don't know anything because it wasn't monitored. But think about it, temperature is one of the most important times. Sleep is extremely sensitive to temperature. And you are not able to go into REM sleep even when you're slightly outside that regular. So cold night and like crap the next day, that's because you didn't get any REM sleep, and that's because you were just a little bit too cold. And that inability to go into REM sleep is going to be even more sensitive to a very small organism like a human infant in these terrible situations, probably with a lasting heat device in the room that probably gets too hot half the time and too cold the other half of the time, which is going to turn these kids into, unfortunately, chronically sleep-restricted and sleep-deprived situation. Probably get lower REM sleep, not going to be twitching, as you can imagine, and they are considered to have autistic-like disorders when they become older kids. So this is just one, graphic example of what can happen, but of course more subtle things can happen as well. So just to conclude, we're getting to a point now where we're recording from enough different brain areas, maybe 8 or 10 or so, some of which just are still being processed, but we're to a point where we can actually start to trace a cascade of information flow through the developing brain with every single twitch that we report. So just to put it all together, if this is the time when a twitch occurs, we're recording from the red nucleus, we will see a burst of activity just before the twitch. And so this shows that the red nucleus is a motor system generating the twitch. And then we can look at the red nucleus, the reafferent processing of the red nucleus after the twitch. We also have information from the external QPH showing that it has a latency that's just a little bit shorter than the red nucleus, sitting between the other two, and that the flow through the cortex is a little bit later. So information is flowing through, cascading through the nervous system because of this system. So we've come a long way since thinking about the system like this, which is really where we began about 10 years ago, to a place now where we can think about the organization of multiple sensory motor loops. So the red nucleus has a spinal projection, which is going to cause a twitch, and there's going to be a connection that comes back directly to the red nucleus. There's the cerebellum system, which has multiple loops within it. So the red nucleus communicates to the inferior olive to the cerebellum. The cerebellum connects back to the red nucleus. There are also multiple projections to the cerebellum, which then feed back to the red nucleus. There's also another loop that goes through the thalamus all the way up to the somatosensory cortex, back down to the red nucleus. And there is a connection to the hippocampus from cortex. This is a projected or a thought about idea that the hippocampus also projects to the red nucleus, but we know that the red nucleus is modulating hippocampus activity as well. So we have loops within loops within loops, almost like what we saw with the spatial organization of the twitches themselves. So twitches again represent a distinct passive movement, and I want to leave you just by saying that our overarching perspective is this. We are not born with some innate of our limbs, their parts, how they work together, but we have to through millions of tiny self-produced movements so that we literally teach ourselves how to map our brain and use our bodies. So with that, I'd like to thank all the previous and present students and collaborators and funding sources, and thank you all for listening.
A really powerful test of your theory, see if there was some way to prevent twitching and look at the effect on developing brain behavior. Any way to try to approach that?
Well, thank you. I'm glad you read the whole slip of paper I slipped into your pocket and feeding me that question. So here's, I've gotten this question a lot. A lot of people suggest, why don't I just put a cast on a limb and stop them from twitching or whatnot. I never did that experiment, even though people kept saying do it, because I didn't see the point of it. Because in that case, you would not be able to discretely affect twitches without affecting weight movements. I felt it would just not be, a very clear experiment. So just last Monday, we received in the mail from Jackson Labs these beautiful new mice. And these mice, we wanted to do optogenetic control of twitching. We didn't know until recently that we could affect the red nucleus and do it that way. That would be really tough to do. Plus, impossible to do developmentally, because the expression times for the virus to do optogenetics is about 14 days. Maybe you can get it down to 10 or 12. That's older right there than the pups that we want to be working with. So these mice are expressing at birth. These are pre-lox mice. They've been engineered to express. Basically, I'll just show you the animal. These are Fiat channel rhodopsin mice. This is a light that's flashing in the spinal cord. Let me just show you that this is not our work, but these are currently mice. Every time the light comes on, you can see that the left hind limb is inhibited from moving. So we think we can do this in infancy. We think we can do this by shining light through the spinal cord itself without having to do it invasively. So we can record it anywhere we want to in the brain. We can do it chronically. We can do it acutely. We'll be able to see what happens when we prevent a twitch from happening while not affecting the while they're going to be, I think it's going to completely revolutionize everything we're doing in our lab right now. Because we can do mice, we've done mice, we've reported in them just like we have in rats. So now we can exactly address the question you're asking and get at the functional features of this and really explore the plasticity consequences of twitching for the nervous system.
I'm wondering if you can talk a little bit about variability in terms. So I'm thinking about it, and I'm thinking about the twitching, and that of course with my own infant, she was doing all twitching and things like that. But then my brother's daughter had no twitching, and also when I tried to explain it to him, he was like, "Well, I think there's something wrong with her."
Yeah.
So we had this big discussion about this. And I'm wondering if you're thinking about individual as a behavior, individual differences as actually mapping out into something. So what?
Yeah. I feel bad for your brother's daughter. No,
No, she's
just
that's
fine.
the thing. Just
She
fine.
moves, but
Yeah.
no, seriously. But we know nothing about twitching. We know nothing about our pups. They don't show a lot of individual differences. They're very small.
Okay.
These are highly inbred animals.
I
I mean, they're born like with clockwork.
Yeah.
But humans are born at all kinds of different times. I mean, there's a huge variability in birth timing, huge variability in developmental rates in humans. And this is also true, I think this is true in dogs as well. I mean, I've seen variability in all the dogs I've had and how much twitching they show. So what I think is there are going to be enormous individual differences. And then the question becomes, are they predictive of anything? This is one reason why I want to move into studying twitching across the lifespan in humans, because we know almost nothing about twitching in humans across the lifespan. Any age. We don't even have quantities of twitching in infants. Nobody's ever studied it because it's been a byproduct. And who wants to spend their life studying a byproduct of a dream, right? Well, if twitching is no longer a byproduct of a dream, then it becomes something substantial. And now it can do a lot of things for you. It can become a sensitive indicator of sensory motor development. It can tell you something perhaps about, it can give you prognostic ability for recovery of function after peripheral injury, after stroke, during neurodegenerative disorders. So you can think about now twitching becomes, and it also can perhaps tell us something about individual differences. It can tell us about skill learning. So if somebody's becoming, learning a new tune on the piano or in humans, in adults, is because the noise band is very high and you have to know what you were looking for. You find that signal in the EEG. It's there. We're looking for it, and we're going to be looking for it. It's there. It's just like an ERP. If you didn't know the ERP signal was there, you can never find it until you trigger it on a stimulus, and you do lots and lots of trials. You trigger on a twitch, I believe that we will start to see that there's a signal in EEG that can tell us something about what the brain's processing very quickly. So I think that there are an untold number of important questions, both medical and non-medical, basic science questions that can be addressed with this. And I think the individual difference there is incredibly small.
It seems very similar to the model of holographic discipline.
retinal waves, but they're really simple waves. The retinal waves are simply a sensory system. And so here we have a sensory motor. So because the retinal waves, there are projections in the retina. They are spontaneous. So the motor event needs to be associated with that particular thing. The level of it is parallel in a way to the story in the visual system, but this far beyond the retina. This has to do with every muscle in our body, and how it gets coordinated in time across all the different sensory motor systems that are involved. So I think parallel system.
And then answer about the temperature dependent.
Yes.
Why?
Well, so don't know. But Evelyn Satinoff did a beautiful study around some movie exhibitions ago, showing that there's an area of thermal neutrality. There's a range of thermal neutrality in rats. These are adult rats, and where they have minimal metabolic rate. Within that range, there's a 4-degree range right there where you can double the amount of REM sleep, around 29 degrees Celsius, which seems pretty warm. At 29 degrees Celsius, you can literally double the amount of REM sleep just by changing the temperature a couple of degrees. So that was a weird finding. It's never really been followed up on, never been tried to explain. The other story is what happens when you keep the animals down enough we can get rid of REM sleep entirely. So the common explanation is that when you are in REM sleep, you actually shut off all of your thermoregulatory effectors. So you are not able to shiver, you're not able to sweat, you're not able to thermoregulate. So the reasons for that are not known. But if you take that as a given, the last thing you'd want to do is to go into a really cold environment and go into REM sleep and then deal with the consequences of being in a cold environment and not being able to thermoregulate. You'd wake up hypothermic or you'd wake up hyperthermic. So the argument is that the gating mechanism is a protection against this design, whatever it is, about REM sleep where you can't thermoregulate.
Do you think, take a moment to listen is pivotal.
It's not pivotal.
But do you think that it's both the comparator and the site of decurrence copy generation?
Well, okay, so we are recording the inferior olive right now. It's really hard, it's really valuable. We have about 4 hits now, and inferior olive, I think I'm probably starting to convince my students that it is receiving a coronary discharge signal from the REM nucleus. It has a completely different shape. It's like it goes this completely low noise, like I've never seen before, and then continues. And it happens about 10 milliseconds after the REM nucleus fires, in terms of what you know about the timing. So we think the inferior olive is getting a coronary discharge signal, but we don't know yet where the comparator is. And so we're chasing it from 2 to the external pineate. So the inferior olive, and we're hoping to converge. One likely spot is the lateral lenticular nucleus, which has been implicated before, but we're getting close.
So this presumably doesn't have an adult. So what's going on there?
Great complicated question. I can tell you when these goes to P12, because we covered that lately. Look at P16, and we think we've seen it there. It may actually go into adults, but let me explain why. What makes our setup good serendipitous is also the same thing that makes it highly unnatural. Because we don't normally twitch in the air. So if you think about it, what's really going on here is that these animals are twitching in an environment where there are no unexpected movements. That's why we were able to do that experimental manipulation. So I think that if we were swimming in the air, or I can actually answer your question a little bit more directly. So when all these movements that I'm making, right? We normally think that the muscle spindles are feeding us back a lot of information. It's actually not true, as it turns out. Muscle spindle information coming back to us is actually suppressed by coronary discharge signals that through it's called the fused motor system. So it's the system that actually controls the motor upload to the muscle. So it's a separate motor system from the one that controls skeletal muscles. And so I thought that we are constantly getting lots of spindle information while we're living, but it turns out that's not really the case, and that's also considered a bit of a mystery in the field. So I think actually there are some legitimate mysteries. It's not as obvious as we thought. And as the more and more I read about this and relate it to what we're doing, it turns out it's just still a lot to some basic stuff we need, basic holes we need.
Do you use it in ethics and adult?
Yes, and people do this. I mean, it's nice to
It's
yes,
pretty common, yeah.
absolutely. I hope somebody.
Okay, my favorite question. Do you think that, assuming your question is right, limited to the motor system, or are there such things as switches in the perceptual system or cognitive system, emotional?
You know, it's a great question. So I'm going to answer it in a brief way, and I'll let you fill in the blanks and see how it applies. So a lot of people think about thought as a form of behavior, and people who have taken these models and thought about things like schizophrenia have kind of come to the same conclusion. So if you think about what happens with a lot of the positive symptoms of schizophrenia, where you have auditory hallucinations, where you have delusions of control, where you have basically trouble delineating the self from other. People have gone in and looked at the function of the correlated discharge system in schizophrenics and found it to be deficient. Now, it's not an easy thing to do in humans, but the psychophysical techniques, psychophysiological techniques to ask and answer that question. So I'm not saying that schizophrenia is entirely, but it is possible that some aspects of the correlated discharge system are dysfunctional in schizophrenics, and that could be contributing to some of these things that seem to fit in with what we think about as a corollary discharge promoting idea, which would mean that if you have a delusion of control, right, then and that confusion between what's happening to you and what's happening and what you're doing yourself, those ideas seem to be compatible. It's this issue of other parts of the brain that are probably depending on corollary discharge systems in order to maintain some synchronous control over what you're doing and what's happening to you.
Just one more question.
So it seems like you've granted a certain special status to the sleep state for organizing the system. At least from the young, I wonder how we now have something changes that occur during sleep and
Yeah.
also related to sleep organization.
I think so. Look, so there's a great experiment that I would love to redo. It was done by Michael Merzenich, where he fused 2 fingers of a monkey and then showed that the map over this course of a month that the map controlling those 2 fingers fused. I would be very surprised if the twitches were not involved in it. In fact, I would argue that if we redid that experiment, you look at the twitches, and you thought about that, because he talks about it in those papers. He talks about it as decorrelating or correlating the information flow. But he's thinking about it in terms of what you're doing while you're awake. When you're asleep, but you know that twitches, fingers twitching independently. And so